We are then ready for our next speaker, Maria Birkisdottir from Erasmus MC in Netherlands. Maria, are you there? Yes, please share your screen whenever you're ready.
Okay, I'm ready. Thank you. So thank you for inviting me to speak here today. I'm very excited about it. My name is Maria Birkisdottir, and I am a PhD student in the group of Jan Hoijmakers, Wilbert Vermeyer, and Dick Jarsma. So I work in the Netherlands, and I work in the department of Erasmus MC and Princess Maxima Center. So my research focuses on how DNA damage, accumulation, and transcriptional stress can drive neurodegeneration, and whether we can use nutritional interventions to slow down accelerated neuroaging. So Jan gave you a presentation this morning about our group, so I will not spend too much time on the introduction, but just to shortly recap, we have here the genomic instability, which is now considered to be one of the main drivers of both cancer and aging. And also now it's emerging as a rival emerging as a driving force of different neurodegenerative disorders. And Jan also showed you the Ercc Delta minus mice, which is a mouse model which is deficient in different DNA repair mechanisms. And it's the most prominently deficient in the nucleotide excision repair pathway. And as a result of this mutation, these mice cannot repair sufficiently or efficiently different DNA repair, both rising from endogenous and exogenous sources. And as a result of that, they start showing very early on this multisystemic degenerative phenotypes. And that is true, especially for the nervous system. But as you can imagine, the neurons are postmitotic and as such rely on a very effective DNA repair for the proper survival. And this is then the case as we've seen in many children with progeria syndrome as well. So we have used these mice a lot, and other deficient DNA repair deficient mouse model to study the role of genomic instability, both in aging and neurodegeneration. But more importantly, they have given us a tool to look at or to screen for interventions that might slow down these processes. And now us and a lot of other people have shown that dietary or caloric restriction has been very beneficial for both health and lifespan, both for wild-type animals, but much more drastically so for these DNA repair deficient mouse models. So what we did in the start was to look at different compounds and nutritional intervention that we know underlie the molecular pathways that are thought to be involved in the reprogramming that dietary restriction does. So I will just spend a very short time on the next slide and tell you about rapamycin, which I've heard has been mentioned a couple of times in this conference. But rapamycin is an immunosuppressant drug, but it's also and works as a large inhibitor of the mTORC growing pathway. And rapamycin has shown to have some anti-aging effect across animals. However, if we try it in our Ercc Delta minus mice, and here again, I can show you that the lifespan of the Ercc Delta minus mice is very short, as they only live between 4 and 6 months of age. And you can see that here in the gray line, that when we put them on diet restriction, you can very largely extend their lifespan. However, if we supplement their food with rapamycin, this doesn't have any effect at all. We then looked at different DNA damage factors, and we don't see any effect of rapamycin on those, but dietary restrictions seem to reduce a lot genotoxic stress in these mice. So we wanted then to focus more on the nervous or the neurological health of these animals. So we decided to look at the Perkinian neurons of the cerebellum, but the Perkinian neurons are very large neurons, one of the largest one of the brain. They are very metabolically active, and they've also shown to be they're vulnerable for both patients with progeria syndrome, but also patients that have different neurodegenerative disorders. And what we did was to basically quantify the loss of the neurons over time in the Ercc Delta minus animals. And we use a method called stereology. So we basically take a systemic sampling across the tissue of interest and randomly sample it within this systemic way. And as a result, we can arrive at an estimated total cells in the whole tissue. So again, here the Perkinian neurons of the cerebellum. So here, this is shown at 16 weeks. You can see the wild-type animals have around 230,000 cells in the cerebellum, or neurons in the cerebellum. But over time, the Ercc Delta minus animals have lost around 100,000 cells. And the same is true for the Rapamycin animals. However, when for those animals that were put on dietary restriction at 8 weeks of age, you can see a large proportion of these neurons are actually safe. So from this study, we concluded that dietary restriction must work, at least in our mouse model, by preventing genotoxic stress. And thereby it can alleviate the detrimental effect that having an impaired DNA repair means. And we can say that rapamycin, again, at least not in our mouse model, cannot by itself have this effect. Furthermore, we can see that the Ercc Delta minus mice show this Perkinian neuron degeneration and that dietary restriction can, up until some point, protect this neuronal loss. So we were more interested in looking at the mode of action on both DNA damage accumulation as a role in neurodegeneration, but also how dietary restriction can have this neuroprotective effect. So as I told you about earlier, the Ercc Delta minus mice have a global mutation. And as such, it's very likely that both cell-intrinsic factors drive this neurodegeneration, but also that the surrounding tissue and the dysfunction of that, along with other systemic factors, can also play a large part in driving this neurodegeneration. And that also then means that we don't know exactly if the dietary restriction is neuroprotective by working on those cell-intrinsic DNA damage factors, or just by maintaining the surrounding glial tissue, or by reducing, for example, inflammation in these mice. So we wanted to answer those questions. And we also wanted to take a look at the effect of dietary restriction over longer time. But as I just showed you, the Ercc Delta minus mice die prematurely. So we decided to make conditional neuron-specific ERCC1 mouse models. And again, we are focusing on the Perkinian neurons. And this is then depicted here. But these conditional mouse models have only the ERCC1 deficiency in the Perkinian cells, where the rest of the body remains healthy, meaning that the Ercc Delta minus mice have this multisystemic degeneration, but here the PCP2 Delta flux mice have the same mutation as the Ercc Delta minus mice, but now only restricted to the Perkinian neurons. If we then look at serology again, you can see the wild type remains the same amount of cells, at least up until 40 weeks of age, whereas the Ercc Delta minus mice show this decline up until their premature death. And both of the cerebellar mutants show a very cell-specific neurodegeneration over time. So if we focus more on characterizing these Delta flux mice, I first wanted to just show you a staining of the Perkinian neurons, but here we have calbinding. This is an antibody that stains specifically for Perkinian neurons. And here you have the wild type situation with the magnification on here of the Perkinian layer. So you can see the dense layer here. And at 8 weeks, again, that no degeneration has happened yet. So the degeneration only starts after 8 weeks of age. So already at 26 weeks of age, you start seeing the gaps in the Perkinian layers, whereas at 40 weeks, a large gaps are observed because a lot of the neurons have already been lost at that time point. And this is then accompanied with brain inflammation. And we use MAC2, which is an antibody, and we use that to visualize activated microglia or microgliosis, and GFAP that we use to visualize activated astrocytes. So you can see that also increases over time in these mice. So now we will focus on the 40 weeks mutants. Again, a cal-binding staining showing for the wild-type animal and for the mutant where most of the neurons have been lost. And what's really nice when we work with Perkinian neurons is the fact that they are very responsible for motor movement. So we can use relatively simple tasks, such as the road rot, to estimate their neurological health or the status of the neurological health. And you can see in the mutants that over time, they start falling more easily off the rotor rod, so they can't finish the task, whereas the wild-type animal shows plateau over the whole time. And again, here, the neurological decline in these animals and the microgliosis and astrocytosis. So this has basically shown us up until this time point that DNA damage can drive neurodegeneration in cell-specific manner. But more importantly, we wanted to know if we could delay or prevent this neurodegeneration. That is, whether dietary restriction could have cell-intrinsic power for cell autonomous neurodegeneration. So knowing that a 30% dietary restriction was extremely successful for the global mouse model, we decided to put a subset of these animals on a 30% dietary restriction starting at week 8. So here is an example of an animal on dietary restriction, and you can very easily see the difference here between the Perkinian neuronal amount. Furthermore, if you look now at the rotor rod performance here now in the red, you can see that the animals that were put on dietary restriction performed just as well as the wild-type animals. So this means that dietary restriction can very well preserve motor behavior in these animals. And then if we look at the stereological data, you can see that a part of the neurons that would be lost in the epileptum animals are actually saved by dietary restriction. And at 40 weeks, the dietary restricted animals actually have over 50% more neurons left than the epileptum siblings. And this again is then accompanied with lower inflammation, as shown with microgliosis and astrocytosis. And yes, so here you can also see the fluxed minus mouse model, which I included in the first graph. But this animal does not have ERCC1 production at all in the Perkinian neurons, but still dietary restriction has some effect. And furthermore, for us to be able to compare the effect of dietary restriction across these 3 mouse models that differ in their severity of DNA repair, we calculated the so-called relative rescue. So here you can see, again, for the flux delta cerebellar mutants, that dietary restriction can have an effect on cell-intrinsic driven DNA damage. And which might be a little bit lower actually for the fluxed minus animals. But if you compare it then with the Ercc Delta minus mice, which again show neurodegeneration, which is probably driven both by cell autonomous and non-cell autonomous effects by the degrading tissue around them, you can see the effect of dietary restriction is much larger. So we believe that this is again works somehow, that dietary somehow works by having this anti-genotoxic stress effect, but we don't know exactly yet how that happens. So we are still working a little bit on that. So hopefully we will know more about the fundamental mechanism soon. So I just want to recap, take my conclusions together. So I showed you that when you have a neuron-specific ERCC mouse mutants, you can see a cell-specific neurodegeneration, which equals to 5,700 neurons on average lost per week for the delta minus cerebellar mutant, and 6,600 neurons lost a week with a knockout situation. So I also showed you that 30% dietary restriction can resave a lot of part of the cells that are otherwise lost, meaning that dietary restriction can prevent cell-intrinsic DNA damage-driven neurotoxicity. And this is probably done by reducing genotoxic stress. The fact that we see this much larger effect of dietary restriction in the global Ercc Delta minus mice means that also in addition, dietary restriction probably also partially work by maintaining the glial cells around it and by reducing systemic processes such as inflammation. And with that, I want to end. So I work in Erasmus with Dick Jarsma and Lisanna. She is there in Denmark, so she has a poster. So I recommend everybody to check her out. And then I want to thank also my colleagues in Princess Maxima Center. And thank you for your attention.
Thank you so much. Great talk. We unfortunately are out of time, but I highly recommend going to Slack and ask questions of Maria.